Firstly, our Dean is going to welcome you, so I leave to Consuelo Serres to welcome you.
Thank you, Susana. Good morning, everybody. On behalf of the entire staff and students of the Faculty of Veterinary Medicine of the University Complutense of Madrid, it's my honor and privilege to welcome you to our institution. Today, we are here to celebrate the closing and present the conclusion of the project IMAGE. It's an acronym of Innovate Management of Animal Genetic Resources. It's a European Horizon 2020 project that involves the collaboration of several researchers and institutions, and 4 years of excellent work. The success of the project is the result of the work of all those persons involved. I would like to use this opportunity to congratulate all researchers, collaborators and institutions that have made this project possible. I am proud that we have such a wonderful group of speakers from our institution and other countries, and I am sure it will be an extraordinary day. I would also like to have the opportunity to thank the organizing committee for their fantastic work organizing this conference, especially to Susana and Michèle to invite me to opening the conference. And thank you very much for all speakers who will share their knowledge today. I hope you enjoy attending this meeting and listening to our college. I'm sure it will be a great experience for everybody. And now I'd like to give the floor to the chair of the meeting, Michèle Tixier-Boichard. Thank you very much and have a wonderful day. Thank you.
So thank you, Mrs. the Dean. So just for the public, so apparently it is the first time that a woman is a dean of this veterinary faculty. So it's really our day. So thank you for being here today. So I will briefly introduce the project and the objective of this conference. And I will also mention that the talks will be recorded so that you are able to find them later on. So in this introduction, maybe I take this one. Yeah. I just wanted to remind the main challenges for science and society that animal gene banks are contributing to address. So you have erosion of biodiversity, you have a renewal of agricultural systems towards agroecology, supported by FAO, and which calls for a new role of genetic diversity. We also have the Nagoya Protocol for access and benefit sharing from the use of genetic resources. But on the other side, so these are all the external, I would say, drivers, and we have some internal driver with the genomics and a strong technology push in different fields, and we have a range of species, so terrestrial, aquatic, with a range of biology, different biological systems to know. So all this calls for an upgrading of gene banking for domestic animals. And in IMAGE, so the objectives were to enhance the use of genetic collections, to upgrade the gene bank management, and to demonstrate the benefits of the animal gene banks. And this will be done. We have done this. We will illustrate today what has been done through genomics, bioinformatics, and reproductive biotechnologies. We had a multi-actor approach in this project because we had stakeholders, both are project partners and others are in the dialogue forum that will be presented. And it is a multidisciplinary project where we gathered animal breeding and genetics, reproductive physiology, but also sociology, economics, informatics, bioinformatics. And so today we will illustrate some of the main achievements of this project, including the better knowledge of the gene bank collections in Europe, and also a very new approach about how to address cost effectiveness of gene banking, and as well as a new analysis of ethical challenges raised by animal gene banking. Then we will have a session on more reproductive physiology and new tools in reproductive biotechnology to improve or guarantee the reproductive quality of the samples, because we expect a lot from the gene banks. Then after lunch, we'll have a session dealing about the new knowledge that we have extracted from genetic collections by genomics, and then how we can put all this data together in a common portal will be illustrated also. And finally, how to use, once we know them better, we have well characterized what we have, how can we use these resources for a range of applications in breeding. This will be illustrated in the afternoon session. And we will finish by the focus on training component. We are here in the faculty, so of course it is very important to have this training, and it has been very successful in the project. And we will conclude with a discussion with the breeders, what can be done in collaboration with breeders, and also what can be extended in terms of challenges for genetic diversity in agriculture, including connections with plants and forestry, because they share similar challenges, issues that we have. So this was just a few words of introduction, and so now I will hand to Hans, who is chairing this first session, and I will open the first talk. And okay, so I will call Coralie. Huh?
Coralie.
And
so, okay. As you have, the title of this sub-session, our first session, is Current Analysis of Animal Gene Banks in Europe and Options for Optimization. There's two talks. The first one is given by Coralie, and I will just be the timekeeper and tell you when to stop if you don't stop in time.
Thank you. Thank you very much. So, yes, I'm not Grégoire Leroy, I'm Coralie Danchin. The slides were prepared by FAO, but FAO, being paid by our taxes, they have a very difficult travel arrangement. They pretty much need the signature of all the presidents to be able to travel. So I think since it was involving biodiversity, Borsalino's one was probably missing, so they couldn't make it, unfortunately. So, I'm going too fast. There. Since you're here, I guess you know that genetic diversity is important. I'm not going to go back to that. Another thing, though, is you need to have in mind that there is about 15,000 breeds that are recorded in DADIS, and that are studied as being national breed populations. So to be clear there, a national breed can be breeds that are not native from your country. There is a lot of countries, for instance, that say that the Holstein breed is a national breed because they have a selection program. And then among these breeds, we have what we actually have local breeds. And if you pull all them together, we have about 8,000 breeds from 38 different species. And also, cryoconservation is important, but I guess you know about that since you're here. Also, this is being a UN talk. Obviously, we have to talk about the Sustainable Development Goals. Maybe you're not aware of that if you're not familiar with UN work, but different countries are, the countries that are part of UN are supposed to try to reach these goals, and some of them are linked to farm animal biodiversity. And this is important for you in your country because if you have the feeling that your work could help with these indicators and it's not moving forward, you could actually go to your minister and say, hey, I know we're supposed to improve this. I don't see any improvement. You have commitment. Why don't you fulfill this commitment? I think it's a very strong political message that you can use to try to finance research and project among gene bank and genetic diversity. So there are two of them that we are very interested by, which are the 2.5 number of plant and animal genetic resources for food and agriculture, and then the proportion of local breeds classified as being at risk. So I could probably stop my talk here, and it will be fairly interesting for Image with this wording, and you will have to work with number of local breeds with sufficient material in the gene bank. You have 2 hours to define sufficient. So there we actually went through we used the definition from FAO. And actually, for the ones who were there yesterday, we talk about the guidelines that maybe could be updated thanks to the work from Image. And so we used the FAO definition to say what is sufficient. One of the main issues, though, when we started the work is that, well, not all the breeds are reported in DADIS, and among the breeds that are reported, there is actually very few data about what is really in gene banks. It doesn't mean that things are not in the gene bank, it's just that FAO cannot know about it because the countries are not reporting what are in the gene bank. And also, one of the issues we saw is that, so there is a big difference between the total number of breeds and what are called as national breeds, and we wanted to try to investigate the case of transboundary breeds. Transboundary breeds in a wide sense, which means that Holstein is a transboundary breed, but more local breeds that share in the Balkan area, for instance, is also a transboundary breed. So what we wanted to try was to assess the effectiveness, gap, and redundancy in the collection of cryopreservation material. We did that for mostly Europe and 2 other countries, thanks to DADIS information, and also thanks to the survey that was done during the Image project, and actually the survey was done mainly for Europe, but then Paul Butcher from FAO actually used that survey to try to have more information from other countries. And there was 2 countries that provided enough data so we could gather everything. In the end, we had 42 gene banks from 15 European countries. There is 1 country with more than 20 gene banks, and we are in this country, it's Spain. We decided to work only on 11 different species. We excluded white species because often we could find, for instance, that there is some giraffe material in one country for some reason and things like that. That was not very interesting. We didn't put fish because fish, there is always difficult status in FAO. It's not with us, it's a different commission. Some countries are taking it to account, others are not. And we're not taking into account pets because there is really, really few countries that integrate pets in their gene banks. Usually it's more dealt by veterinarian faculties and not in the agronomy environment. So in that we had almost 700 national breed population, including almost 400 local breeds and 230 transboundary breeds. And what is stored is mostly semen. There is a little bit of embryo and then other stuff, but the other stuff is less than 1%. So after that, we tried to have to categorize them according to different status. Very easy. Sufficient material is it equal or more than the threshold that was defined by FAO? And you have to keep in mind that the threshold differs between species. Insufficient or no material. No material was when there was a breed reported in DADIS and we didn't find the breed in the gene bank. But we did surveys, and as you know, surveys, the quality depends on if the people are actually participating in the survey. So you may say, hey, but we actually have this breed in my country in the gene bank. Yeah, but if it's not in the survey, we don't know. So obviously there is some gaps in that too, because we don't always have the information. So this is all the countries. So you'll see better when you have the slide that will be available. But main messages, gray is one there is no material. So what you can see is that pretty much the slide is gray, okay? Also, there are large difference between countries, and the countries that have the most material are Austria, France, Iceland, the Netherlands, and Spain. Good, the meeting is in Spain, where there is the larger proportion of the national breed population with material collected. Every time you will see the acronym NBP, it's National Breed Population. If you go, when you pool then the number by breeds, which means, for example, there in the previous graph, the Holstein was counted in maybe 20 times in each gene bank. There we are pooling. The Holstein count only for 1. Well, in the end, we pretty much have the same results. And what's important is that the species with the most material are ruminants and pigs. And then, oh, there is S, that's not there. Donkey and horses, and then over species. And it's only in cattle that you will have more than 10% of the breed with sufficient material. So this follows a bit the economic power of each species, but not only. And we'll go back to that in the conclusion part. If you look that according to the geographical status of the breed, which means international breeds, regional or local, if you monitor by counting by NBP, there are no differences. However, if you count by breed, there you can see differences, and that's what's important, is that the transboundary breeds start to have more material. Also, if you look at breeds according to their risk status, well, that's kind of unfortunate, but the breeds that have the most material are not the ones who are endangered. They are the international breeds. So, the take-home messages, frozen semen is by far the main material collected, but that has to be linked to this part, which is the development, the reproduction technology. It's the technology that's easiest, the cheapest for most species. Also, we have to keep in mind that there is an important difference between countries as far as cryopreservation efforts. And there we're talking about gene bank like it's not, we're not counting AI center, okay? We're actually trying to count the countries that say this material is meant to be kept for long-term purposes. It's not a commercial. If we look at the status and we merge the breeds together, it actually improves the cryoconservation status, but this improvement in a way is not done, it's not coming from a collective effort. There isn't a collective effort to try to have all the genetic variability of the Holstein between countries. It just happened that everybody has Holstein in its gene bank, and maybe we could improve there by seeing if we are not storing, if everybody is not storing the same thing. Also, the breeds that are not at risk are the ones who actually have the most material. Well, that goes back to the fact that there are a lot of time, there are also transboundary breeds. Also, there are more animals, so it's easier to access the animals, because a lot of time one of the issue is, okay, you want to collect a breed, but just the logistic of taking a male and bringing it to an AI center sometimes can be extremely difficult. So, of course, the more males you have, the easiest. And a lot of time also is that in these cases, what happened mostly in cattle, for instance, is that at first the bulls were collected not for the gene bank. It was collected, for instance, for commercial purposes. And then since in a bull you can have a lot of doses in an easy way, it was easy for them to give some doses to a gene bank after. I mean, when you're collecting, for instance, there is a bull that had 1.6 million doses. If you say, can I have 200 for my gene bank, it's not a problem. For a rooster, when you have 40 doses, you're happy. So it's not the same there. Well, take-home message too is that you should go to your Minister of Agriculture or Environment and say, I'm sure we can do something more about gene bank, pretty much all countries, because the local breeds a lot of time are not well cryopreserved. There is still work to be done. I think we can have image number 2 and number 3 and number 4. We have work for the next 20 years. Even if Michèle at one point will have to retire, we'll have to have someone to take over. 20 years, I think. And then, yes, of course, we need better coordination among gene banks at national and international level. So at European level, we're trying to do it through the UGINA network and ERFP, and there it's the same. We still have worked for quite a bit of time. And maybe one of the issue there is, and I think it's image we found, we realized that, and now we start to say more and more often, is that it's easier to convince people that you can store material if you can show them all the different uses you can have. And to show the uses, you need to have better information on the sample, which means you can say, for instance, okay, I have sex semen. For instance, you can say, oh, okay, I have actually the diversity of the breed, and I'm certain because I did the molecular data, and I make sure that they are all not related, or I know that this one is carrier of a gene that's very interesting, or things like that. And also, one of the main points, if there are some national coordinators here, is that obviously there is great need to have the data that be gene bank data sent to DADIS to improve the FAO data. And that's also one of the major work that we have to do with the help of ERP and UGINA network. And thank you for your attention.
Does seem to be working. I will shout. Thank you very much, Coralie. Thanks also for keeping time. It made my task easy. Any questions? Maybe I can start. So when you introduced, you said you also looked into transboundary breeds that are close to each other and just a few, 2 or 3 countries involved. I didn't see much coming up on this one, or maybe I missed this one. What kind of experience did you get for this one?
Well, the transboundary is the difference between national local breed, breed population and breed. And transboundary can be in the wide sense. I mean, Holstein is a transboundary. Yeah. And what we found is that when you go from national breed population to breed, the status of breed on some of the breed actually improves as far as cryoconservation. But it improves more for the big international breeds than for the local, like the example of the Balkan sheep, or I'm not sure, I didn't look closely, but I'm not sure it improved a lot when we actually pulled everything, because I'm not sure there is any Pramenka sheep in a gene bank, or probably in Slovenia, but I'm not sure in other gene banks. So in a way, it improves for the richest breed.
I would be interested in your suggestion. What should we do with disease carrier? Should we also conserve that? They can be interesting for genetic diversity perspective, but on the other hand, they can also cause problems in the population.
You mean, for example, sheep that are sensitive to scrappy? Yeah. I mean, I think that's mostly... Identify carriers of the disease. Yes, I think this is really depends on each country. I know in France, for instance, we have quite a lot of... we have a fairly big sheep collection thanks to that, because all the carrier, but at a good genetic level, they wanted to get rid of it. And we thought it will be maybe stupid to get rid of a good ram just because he's a carrier if the semen was there, because already collecting a ram costs quite a bit. So we have it. And when it's a simple gene, I mean, you just need to, if we want to use it, we just need to breed with a female that's not carrier, and And the offspring, I mean, we have, the odds are fairly high that we'll have an offspring that won't be a carrier. So, but I think that's more a country decision.
Yes, please.
If I understand well, that means what you said is that the Center for Natural Resources made not always the right decision to see what is stored, because the transboundary breeds have been stored quite nice, but the local or the native breeds very poorly.
Yes, in general.
And what would you suggest that there would be more emphasis on that, or it's...
Yes, I would suggest that, but also, I mean, with the work we did also with the equivalent of Image in France, realized that when a breed is really close from extinction, you need to think twice about collecting, because sometimes it's so inbred that already it's difficult to have semen that's fertile, and when you do the AI, you realize you have no results. So I think it's a very practical thing, but in a way where message now is more, you need to focus on your local breeds that are not completely inbred yet. Because there you will still have diversity, there are good chances that you can have good quality animals, a lot of samples, and you'll be efficient there. And I think it's, when they had the seminar in Morocco a couple of weeks ago, they were talking about that, say that there was a breed in Morocco that there was a lot of animals like 10 years ago, almost now there isn't that many, and they are thinking of the cryoconservation now, and in a way it should have done it when the population was not at risk, and it's still a local population, you know. So the message is kind of shifting, well, from our point of view, we're kind of shifting for, okay, extremely rare breed, think twice, or maybe very specific material like oocytes or things like that. But if you want to make, if you don't have that much money and you need to make choices, maybe you should go to the local ones, because there you know you'll be efficient.
Okay, thank you very much indeed. The last sentence brought up exactly the next topic, which is actually about economics and choosing between and so on. And Rafael, you can talk, please.
Okay, thanks. Okay, so thanks for the opportunity. My name is Rafael Silva, and I work at the Global Academy of Agriculture and Food Security, University of Edinburgh, with my colleague, Professor Dominique Moran. And I'm going to present these rationalization problems that I have worked with him, Oscar Cortes from here, and other colleagues as well, right? And then I'm going to show you two cases of rationalization, because when you say rationalization, what do you mean? You need to define what is your problem, what are the research questions you are trying to respond. So in application one at the European level, we were looking at the question asking, okay, given the current collections in the gene banks in EU, we ask, is it optimal? If it was possible to reset the collections, would be saving money or not, right? And the application two is a bit different. We're actually planning future collections for Spanish livestock breeds based on in situ projections from today to 40 years or so in the future, all right? So just to go, to have a look at a bit of context. So basically you have two approaches for conserving breeds, in situ or ex situ, but the problem with in situ is that animals are somehow vulnerable, for example, to climate change, and there is homogenization of the breeds in situ sites. Recently there has been an increase in interest in monitoring, so more data for us to analyze. For example, the second report, FAO report on animal genetic resources for food and agriculture. If you look at the literature on in situ, ex situ, most of the studies are actually focused on in situ rather than ex situ, and the ones in ex situ, they're looking at genetic information and running optimization algorithms to rank the breeds. And the problem of rationalization of the collections in terms of the logistical problems has been a gap in the literature that we are addressing. And then why? Okay, so if you have, this is a problem, it boils down to the problem of having limited resources, right? If you have unlimited resources, then your problem is solved. You collect all the breeds, and that's it. But ex situ conservation is expensive because of need for special equipment, techniques, trained staff, and cryotanks, and so on. And then, this is the first case. And then I'm going to present this case that we published a paper last year for the European level, right? And we try to identify what are the economically efficient rationalization for the gene banks, exactly with that problem of accepting that resources are limited, and we consider collective objectives across 12 European gene banks. And then what are the questions that we're trying to respond? What are the questions we're using the optimization model to respond? For example, how many doses of genetic material, how many doses, semen doses to collect? Each bank should collect, where to store, what are the breeds, and what would be the costs of it. And then to do that, we developed this optimization model, mixed integer programming model. First of all, to estimate the current cost of the current allocation, to identify overlappings in the gene banks, and to identify the optimal collection and storage in the cryobanks, optimal minimum cost. And we used data that we applied a survey for economic data and getting capacity of gene banks and so on. That was conducted in 2017, the IMAGE project. And then, in short, the model finds the most cost-effective collection and storage strategy, allowing for cross-country collections. Some of the constraints, right? The regional availability of the breed in the regions, collection cost, the distance from the gene banks and collection regions, and capacity of cryotanks. And then the model will tell us how many doses each gene bank should collect of livestock breeds, so that costs are minimized. Then here's an example. This is just an illustration of the model, right? Imagine that you have 3 gene banks. Then let's say gene bank 1 has the decision of collecting breeds in a region 1, or gene bank 1 can go to region 2 or region 3 with different distances and costs, and so on for all the 12 gene banks. Then as an example of solution, it could be something like this. Gene bank 3 collects 100 doses of sheep Limousin from region 1, 300 doses of cattle Aberdeen Angus from region 2, and 150 doses of pig Basque from region 3, for example. Okay? And then a bit of the cost data we use the survey to collect. It includes technical coefficients and costs. For example, the cost of semen freezing, labor, documentation, other collection costs, cost of skilled labor, material and equipment. And of course, the information of the current collections in the gene banks. And here's a summary of some of the information for the 12 gene banks, locations, the maintenance costs in euro per dose, tanks capacities in terms of semen doses. This is 0.25 milliliters. The number of current doses in the gene banks, travel cost, and average distances to the collection regions, farm regions, right? And here it's the current allocation of semen doses in number of doses across the 12 gene banks and livestock species. And then we have a total of 517 breeds with around 1.5 million doses. You can see most of the doses are for cattle, sheep, and then pig, goat, horse, and poultry. And then the idea is to reconfigure this and ask how much could be saved in terms of money. We identified considerable overlapping in the gene banks. You can see all those breeds have doses in more than one gene bank. Of course, some of them are transboundary breeds, but some are not. And here I'm showing you the current allocation of the gene banks. And for this allocation, it costs around 22.3 billion euros. And this is suboptimal. If we press a button, like if it would be possible to reset the collections, we could save around 23% of the total cost. That would drop to 17.8 million. So you can see a change here. So the model is telling us that gene bank 1 is more efficient. Gene bank 2, it suggests us to not collect at all in gene bank 3 based on efficiency, cost, distances, etc. And this is how you can save money. And then the other question we try to respond is, what if instead of having the collective 12 gene banks, we concentrate all the collections in just one gene bank? And we found that it would not be a good idea. So this is the current collections, around 20 million euros I showed. And this is putting all the collections in gene bank 1, in gene bank 2, gene bank 3, and so on. You can see that it's not a good option. Even if we put in all the collections in the most efficient gene bank, it would still be nearly the double of the optimal solution, where we are distributing the collections across all the 12 gene banks. And then another thing we looked at is the sensitivity analysis of costs as a function of number of breeds collected. So this diversity here we're defining as the number of breeds. And this is telling us that in the beginning it's cheaper to collect the breeds. So with small variations on the budget, it's possible to increase a lot the number of breeds, up to a point it kinds of, you need more and more money to collect more breeds, because the model is going to tell you to go for the more expensive breeds and to regions that are more expensive to collect, and to add doses in gene banks that are less efficient because of limited capacities. And then we go to the conclusions. There are costly overlaps. This is the kind of thing you want to avoid when optimizing, rationalizing collections. There is a potential cost saving, as I said, around 23%. Centralizing the collections in one gene bank is not a good idea. And yes, of course, this is sensitivity analysis, shows that the costs are not fixed. They depend on the target diversity. And yeah, there are numerous challenges for rationalizing collections, institutional, economical challenges, but they can be informed by mathematical modeling, like this example with optimization I showed. And then we go to the second case about this Spanish gene banks, and I'm going to invite Oscar to give a bit of context. Then I come back.
So in the second part of this presentation, we are going to show you the results of the analysis based on Spanish data. And the title is Rationalizing ex situ collection of Spanish endangered livestock breeds. And in a few slides, I would like to introduce you some particularities of Spanish local breeds in order to a better understanding of the results that Rafael will show us later. So from 1930 to 1970, and as a consequence of different circumstances, the Spanish local breeds were replaced by autochthonous breeds, by commercial breeds, more productive, and many of them were at risk of extinction due to the dramatically decrease of their census. So this is a worldwide problem, as we can see in the different reports published by the FAO. So the national authorities and also local authorities launch different actions in order to preserve the genetic resources of the local breeds. These actions had their continuity in a royal decree published in 2008 by the Ministry of Agriculture that includes different topics or different actions in order to preserve the local genetic resources. In spite of all these actions have had good results, we need to still working on it, because the majority of the local breeds in Spain are classified as endangered based on census data. So, as I said before, the Royal Decree published in 2008 includes different topics as ex situ actions just to strengthen the development of the gene banks. And Spain is organizing different autonomous communities. 8 of them have a local gene bank of the autonomous community, and the main goal of these gene banks are to store biological material, mainly semen samples, of those breeds located in the same autonomous community, but also of transboundary breeds or other breeds located, other local breeds located in other autonomous community. Also, there are public research institutions and universities that collaborate in different projects, and part of the research activities of the projects is just to collect samples, different biological samples of local breeds, and at the end of the research activities, these biological samples are stored in the institutions, and also there are private companies that sell embryos, semen samples for local breeds and also for transboundary breeds. Many of these institutions have collaborated in mass projects, answering different surveys. Some of the managers are here in this room. I would like to thank their collaboration to all of them. And finally, there is a national gene bank located in Madrid. The main goal, or this national gene bank, is a permanent collection. It's like a backup of the biological samples of all the local breeds. So the local national gene banks and also the institutions send to the national gene banks sample of the local breeds to store in this national gene bank to be a permanent collection of local breeds. So now Rafael will show you the results of this analysis.
Okay, so continuing to this second application. In this second case, rationalization is about planning future collections under extinction risks. So based on extinction risks, we also want to identify still how many doses of semen to collect, where to collect, when to collect. It's the new question now in comparison to the first case, to minimize costs. And it's the same idea as the first paper, to identify economically efficient way of collecting livestock breeds, endangered breeds. And in this case, we're also exploring the trade-offs with in situ populations. And then this time we used a stochastic linear programming model to identify optimal collections, subject also to geographic distribution of the breeds. We used census data, extinction risks that we calculate for future, cryotanks capacity and other logistical constraints. And here is the map of the 18 gene banks and the central backup gene bank that Oscar was saying. So basically, of all the new collections, half of the collections are going to be allocated in the backup gene bank. And then we used data of costs. so economic data for costs of staff, collections, maintenance, capacities, and information on the breeds, number of semen doses, and costs, and how many donors, etc. And we use census data that use data from 2009 to 2018. So I have numbers of male, females, total number of animals for cattle, sheep, pig, goat, horse, and chicken. And here is a summary of the cost data of the 18 gene banks, location, right, the capacity, 0.25 milliliters of semen doses. This is a fixed cost in euro per year, and approximated collection cost in euros per dose. And here is just an example of the census data. So we used actually 180 livestock breeds. Here I'm showing you the example for one breed, right? So I have from 2009 to 2018, males, females, etc. And we actually worked to simplify, we used the total number of the population instead of proportion of males and females. And then you might ask, okay, but what it means extinction for you? First of all, before rationalizing, we have to define it. And then we used FAO guidelines when you should intensify ex situ. So basically, we worked with two scenarios. One, when the population size or population status is less than 1,000, and then we are considered under risk of extinction when it's below 500 animals. Two scenarios. And how did we work with the projections? Use the census data, right? So imagine that those points are population size from 2008 to 2018, 2009 from 2018. We don't know in the future. It could be anything, right? But we did a regression, and we calculated the uncertainty bound. So let's say we're here, 2020, and here is 2040. We don't know where the population is going to be, but probably it's going to be between the lower bound and the upper bound of the linear regression we used. And how do you know what is the probability of a breed being at risk or being below N, 500 or 1,000? Well, it's basically this distance divided by this distance. So this is how we're calculating the extinction probability. So we have data for each from 2018 to 2060 for all the 180 livestock breeds. Then another variable that we use to rationalize future collection is, okay, you have the extinction risks calculated from the projections, and you have what we called the accepted level of risk. So what it means what are the risks you're accepting to run. Let's say you're gonna, because costs are very high, limited budget, and then say, I'm only gonna bother collecting a breed that will present at least 25% of being extinct, right? Or 50%, it depends, 75% and so on. So we use that data, and then we compare with the projections that I showed in the previous slides. We put that in the optimization model that will determine the decision variables for the collections, doses, locations, when to collect, etc. And so that this is met. So you're collecting the breeds that will respect the acceptable levels of risk. And then the model will tell us the minimum cost collections allocations, and we also derive the efficient cost frontier of risk versus cost. And this is the projections based on the census data. So each of these curves are showing you the number of livestock breeds under, so orange, for example, the number of breeds under 5% extinction risk. So dark blue is the number of livestock breeds that will present at least a 50% of being extinct from 2009 to 2060. And this is considering extinct less than 500, and this is less than 1,000 animals. Some of the information you can extract from these figures are around 70% of the livestock breeds in Spain are already below 500, so it's the gray curve here currently. So if no ex-situ actions are taken, then around 25% of the Spanish livestock breeds will be under 50% probability of being extinct between 2040 and 2060. Around 15% of the breeds will have a 75% risk to be at risk by 2040, for example. And around 35% of the breeds will have a 25% of extinction risk from 2030. Just some highlights of the information you can extract from here and here. And here we go to the cost analysis, the efficient cost curves. And then here I'm showing the decision variable of probability of extinction in accepted level, you're supposed to, you're accepting to run, and the total cost in euros per year according to livestock species, right? And then we found that the total costs in Spain varies from 95,000 euros per year to 173, and from 115,000 euros to nearly 2,000 euros per year for the scenario of n equal 1,000. This is more expensive because, of course, this is 1,000, this is 500 animals. And you can see the interesting thing here is that the costs actually doesn't change a lot. So it doesn't matter if you're being conservative or not. Costs-wise, it's not going to change. And also, if you look here, for example, you can see that, yes, if you're very conservative, you have a lot of money to spend, and you say, I'm going to collect all the breeds, if they have any probability of being extinct, 5%, I'm still going to collect. Then the costs are going to be higher. But as you relax your acceptable level of risk, then costs are going to reduce, of course, because there'll be less animals to collect, up to a threshold point where the costs no longer change. So this is like the optimal point to act. This is important for policymakers, for example. And the interesting thing is that you can see that most of the costs are for sheep, then pig, then chicken, and horse. But if you look at the data, cattle breeds are more endangered. But the thing is, because most of the cattle breeds are already in the gene bank, so the model knows that, and we only collect the breeds so that it guarantees the minimum number of doses, and therefore it suggests to direct the cost to other species than not cattle. And then I'm just showing you the comparison of the number of breeds currently stored in the gene banks, 2018, and for example, 2040, assuming that collections only for breeds that will present at least 50% risk of extinction. And then I go to the conclusions. There are significant extinction risks for many Spanish breeds. The uncertainty analysis reveals that ex-situ costs are projected to increase over time. Particular attention must be given to sheep, goat, and pig breeds, as I was saying. And intensifying ex-situ efforts to 500, considering 500 or 1,000 animals does not change costs significantly. And costs are very sensitive to accepted endangered levels up to a threshold point where they remain the same. And another conclusion is that the current capacity of the gene banks will sufficient to 2060. There's no need to acquire more cryotanks, at least, if you're collectively distributing the collections in the 18 Spanish gene banks. And yeah, so acknowledgement, thank you very much for gene banks that gave us the date and Emerge project and the founders. And thanks for your attention.
Please come up as well for discussion.
So I stay here for questions.
Yeah, please, both of you. Any questions? Maybe I start again. Your projection of risk, isn't that very risky or very vague because it's depending so much on politics? Well,
the thing is, thanks. Yeah. So the thing is, it's a projection. So when you take the census data, right, from 2009 to 2018, and you do a projection, you're basically assuming that in situ will remain the same. They're not going to change a lot. And that's,
so that's kind of a...
Yeah, it's assuming business as usual for in situ projections.
More questions? Sipke?
I think in many countries there are examples that we regret that we did not collect samples in the time when the breed was not endangered yet. So shouldn't we just promote that we should regularly make a backup of any breed? Because again, it's not so easy to predict extinction probability, right?
Mm-hmm.
Looking back, we have many examples that we regret that we did not sample enough when the breed was still not extinct.
Yeah, I agree. I mean, that's the main thing, is that you have to act under uncertainty. And that's why we derived this model where you consider uncertainties, but not just the projection. The model also looks at the current doses. And let's say there is a minimum recommended dose number of 3,000 doses of semen for cattle. And if there's 2,000, the model will also tell you, okay, the breed is already there, but there's not enough doses, then collect more. And that will be included in the cost estimate.
There's one more question here. Please.
Thank you for your presentation. It's really very interesting. And I would like to have a copy, if possible, because now we are working in the Ministry of Agriculture to prepare the future budget. So it will be very interesting for us to support our request to have more budget for these projects. So the point is, what do you feel that what we could do to create awareness to convince all the governments, all the stakeholders, all the people about the necessity for the future to allocate more money, more budget to conserve these breeds. What do you think about that?
Well, I think exactly one thing is to show the projections that indeed lots of breeds are going to be under risk in the near future. Like nearly 30% of the livestock breeds are already under extinction risk, less than 500, right? And a cost analysis like this showing, because this is what policy makers, what they want to know. It's all about costs, right? Costs and the risks you are accepting to run. So if you show then like a picture, okay, this is the probability of extinction, this is the accepted level of extinction, and this is going to cost this. If you're not concerned about breeds up to this level of 50% of extinction, if you're only investing money, if it's 75% risk of being extinct, the costs are not going to change. So you have a way to convince them that's more sensible to collect right now instead of waiting when the risks are going to be higher. So that's the purpose, and I'm happy to share anything, the paper and the results with you, because that's what we are trying to do to influence policy in the end.
You would like to add this one?
These are the preliminary results, so also we will send you all the results later, but we have this idea just to send all to you these results of this analysis.
Okay. I think I have to close right now your presentation. Thank you very much, all of you, for presenting. And the last speaker is Michèle, talking about ethics.
Okay, so good morning again. So now I will talk to you of a very complementary approach. You have economics, and then you have society and ethics. And if possible, they have to agree and go together. And this is not always spontaneous, I would say. And so I would like first to acknowledge my co-authors, because I am not a specialist in ethics or in sociology, I must say. So Antoine Doré is a researcher in social science from INRAE, and Denis Laloué and Éléonore Charvolin-Lemaire did the analysis of the ethical survey, and Waltraud Kogler organized the dialogue forum. So I will be presenting what they have done, actually, what they have organized, and what lessons we can obtain from the different approaches we took. So biobanks are at the crossroads. So I use biobank, which is a generic term. It includes both animal gene banks conserving reproductive material, the ones you have been hearing a lot since this morning. And the aim is to produce live animals, and the users are mainly farmers, breeders. But we also have biobanks conserving biological material for genomics, for research, with slightly different type of users. But these are very complementary. So they were both included into the objectives of IMAGE project. And if we look at the decision makers, or the social arenas, as you could say, then Antoine has drawn this nice scheme adapted from these authors, published in 2012. So biobanks or gene banks, they depend on legal decisions or legal framework. They also depend on the trends in scientific domain, in technology, also in trends in industrial, in the economic world, and also in trends for agriculture and breeding methods and knowledge. All these different drivers or decision makers are different, and the intersection of these four domains create a sort of what we can call a socio-political arena, where you need to have this a consensus or agreement regarding optimization of the management of gene banks. So we try to approach this complexity with the three methods. So we set up in IMAGE a dialogue forum, which is a multi-actor structure with a yearly meeting. So we had 4 meetings of the dialogue forum, so one topic for each meeting. And in this dialogue forum, we had policymakers, so mainly from agriculture ministries, gene bank managers, scientists, and NGOs, and a few breeding companies. They were generally sending one representative for all of them. Then we conducted surveys, different types of surveys, and particularly one regarding the expectations from stakeholders, and a specific one for ethical considerations that I will detail a bit more. And there were also colleagues conducted interviews, directed interviews or semi-directed interviews, because in a survey, you know, you have to tick the answer you prefer. In an interview, you provide much more details or nuance in what you think, but then you cannot do as many interviews as we could get information from a survey. So the dialogue forum, here are the 4 topics which were addressed in each year. So the first one was really the launching about the main priorities and perspectives for farm animal gene banks, getting the views from the different members of the dialogue forum. And one message was that there was generally a positive attitude of the different stakeholders regarding the role of gene banks for animal genetic resources, that they can really provide a useful support to the conservation of genetic diversity in the livestock sector. I think this was already good to have some consensus among different stakeholders, at least on this positive view. Then in Tallinn 2017, the focus was about sanitary regulations, which address actually one of the previous questions about disease carriers. Because, well, we are not going to store in a gene bank infectious material, or if we have to do it because there is no other way, it has to be in a special place, completely separated, with a lot of documentation, and generally this is not done. But there are very strict animal health regulations motivated by international trade. So they are very severe, and many of us consider they are too much severe for local breed management. The goal is not international trade for the management of a local breed. So some disease monitoring programs are too much severe. Local breeds cannot afford them, and then they are not eligible into gene banks just because of this. So this was discussed at this dialogue forum. How could we still stimulate exchange or collection of gene bank material for breeding and science, taking into consideration health risk in a more practical way, not only international trade driven. And so finally, there was a consensus to propose to recognize the specific issues for animal gene banks in the European regulation for animal health. That's what is called a delegate act for germplasm, because we discovered that in the world of policymakers for animal health, generally most of these policymakers are unaware of gene banks and preservation of genetic resources, so that there are contradictory policies. The animal health policy is not going in the same direction as the genetic diversity policy. And we have a priority of breeds, for instance, for the agriculture policy, and then when we have the animal health priority, we cannot do anything for these breeds. So, at least what is now written in the Animal Health EU Delegate Act, that countries are allowed to take special measures for gene banks. It doesn't mean that they have to do anything, but they can, because usually, at least in France, they say, oh, this is the European regulation, we cannot do anything. Now, in the European regulation, it is written that a country can justify a specific measure for this or that objective of genetic diversity, and we hope it will help the cooperation between neighboring countries for local transboundary breeds, because each time you cross a border you are international trade, and then animal health regulation were very strict and sometimes were limiting the cooperation between gene banks for one local breed. So I think this dialogue forum has had a real impact on both the regulation and the practice, hopefully. In the third dialogue forum, there was the presentation of the first talk, previous talk, about the global economic rationalization of gene banking. And I think in that forum it was really raising awareness. It was sort of discovering that you can have this sort of approach, because it was never discussed before. And of course it was not immediately understood or taken as a regular practice, but I think it was very important to make the gene bank managers realize that they can use this sort of approach to see their strategy or revise their strategy. And the last dialogue forum was, well, in a place I cannot pronounce the name, in Belgium, I would say, organized by SAVE with to have more NGOs as usual in the dialogue forum. And then it was when Ella Martiniuk made a marathon, I would say, about the Nagoya Protocol, because it is a very complex legal framework, and so this dialogue forum was really a keystone to describe all this new background about the Nagoya Protocol, what are the key issues, the impacts, is it really changing our work or not? And all these, there were two presentations. They are on public access on the website of IMAGE. So I will not tell it again, because it is a very complex framework, but I think it was aimed to help all the gene bank managers and users of gene banks to get a reference document of what is this Nagoya Protocol. So please go to the website and you can download the presentation. So about the ethical issues. So first of all, what is ethics? So already this referred to old notion, 1922, that ethical implication of biobanks, because when you are acting, you need an ethics. If you do not do anything, okay, you don't need to have ethical questions. But ethics is about how to act. And gene banks, they have to take decisions, not alone, with the partners. So finally, ethics is always there. And biobanks, they are collecting material, distributing material. They have a lot of stakeholders. So of course, ethical issues are relevant. But what was a bit, to tell the truth in the story, that we did not put that as an initial objective of IMAGE. It was really a request from the ethical committee of the Commission. When they accepted to fund IMAGE project, they say, okay, yes, but you have to do ethical analysis on the considerations about the choice of breeds to be cryopreserved. What is the opinion, the different opinions of stakeholders? And also because we had a work package working on new technologies for biotechnologies, which may raise issues of acceptance, or maybe some technologies are a bit difficult to understand or to accept. So we had also to consult stakeholders on this. And in parallel, I will show results of interviews. There were 15 interviews conducted with animal biobank managers in Europe and 27 on in situ conservation stakeholders, well, small farmers, but these were only in France. So maybe it's a French story for this, but you will see it is sometimes very interesting to compare general analysis of a survey and individual opinion of a different stakeholder. So the ethical survey was launched in 2018. It was distributed to different people in different events. It was a bit of a challenge to be sure that we could reach as many stakeholders as possible. And it was organized in 3 sections. So first of all, we need to know who is answering. So general information about the profile of the respondent. Then questions about the motivations for conservation of breeds. And a third round of questions about acceptance of technical innovations associated with gene banking. And there were 159 respondents. So here you don't need to see all the details, but just to show that we had different types of institutions, people with different roles, different types of institutions, mainly public, but also a few private, breeding companies, less numerous than breeders' association, a lot of colleagues coming from scientific institutions, and also people from government, so decision makers. And in the role, we had either people working on the technical management or at the direction level or in the scientific world. And we asked the respondent, we didn't ask their name or first name or age, these are private data, but either they expressed their own opinion or the view of their institution. And so we had a few other roles. You always have different answers. So I think one point I would like to stress that we asked them, are you working on rare breeds only, on commercial population only, or on both? And I think it was quite interesting to see that the highest category was working on both. And this is an important message because you have seen this morning we have spoken a lot of rare breeds, and although for commercial transboundary breeds everything is going spontaneously, but then it is important to see that people managing commercial breeds are also involved in the management of local breeds. So they are not completely two worlds, although there are different issues, but the people can be the same, so that it is important to deliver different messages maybe to the same person depending on the role. So we had actually a minority of respondents only engaged into commercial breeds management. And here is I extracted the data just from the French survey, and it was the same, that both or on rare breeds only are big, equivalent categories. And also this was true, this distribution, either for single species or several species. We had half of the respondents working on several species and on rare or commercial breeds. So which showed that at least we don't have too much fragmentation. And now the first question, one important question is what are the objectives? Why do we want to cryoconserve breeds? So again, I have shown here the different profiles of the respondents to remind. And here is, so this is a game sometimes I use in a training session, but here we had no, what do you guess this is this answer? Number 1, the one with the highest answer, as compared to what we heard this morning. Well, this is insurance to revive or restore a lost breed. So this was not a big discovery. Then the second one was supporting in situ conservation. Then the third one was research. So actually we were happy to see that research is not so a minority of objective for cryoconservation. Then introduce diversity in breeding programs. This is introducing diversity in commercial breeding programs. And we can see that they are less likely to use it than supporting in situ of small populations. And the last one, which is always the one I am the most sad about, that new breeds, making new population, mixing genes, having composite breeds, this is always the one getting the smallest number of answers. Maybe only I have one answer out of 5 for me here. So this is, but I think in plant breeding, people are using gene banks much more often to create new genotypes. And in animal gene banks, no, it's not at all viewed as an objective. Maybe it will change, but it's not the current situation. So if we compare that to some, so we have our questions, and one answer that Antoine got in the interview saying, I don't think that the Rare Breed Survival Trust will be able to afford for all the different breeds. So which means that we need to choose. We cannot do all, so that's the goal of the previous presentation. So what are the objectives of the gene bank? A gene bank has to declare its strategy and its objectives because there are so many choices. And so explain the criteria and who is going to prioritize, who is going to decide, who shall pay, and also should there be a balance, is there an ideal balance between ex situ and in situ conservation and support? And at least I can tell you that for these two, it was very clear that those who pay are not those who decide because it has to be a multi-actor decision. Okay, so it's really the question of having public good or club good because this was a very strong message from the answers. It's not only a fund issue. Well, also maybe this is limited because we only had 3 answers out of 159 of people declaring my role is to be a funder. So of course their answers were maybe a bit lost in the whole answers. So the main criteria to choose a breed for conservation, here is the answers from the 159, and here I put the one we had in France. So high degree of endangerment is always the winner. But then on the main survey, we had special traits and characteristics of value, which ranked second, but in France, it was not ranking second. And genetic distinctiveness ranked third in the global survey, and in France it ranked second, very close to cultural heritage, which was 4, so there was some change in ranking, and this one, all breeds should be cryoconserved. It was, well, number 5 ex aequo answer, and in France also. So what is interesting that when you ask these questions, and we could still continue to use these questions in different arenas and compare the profile of answers, because when we were in Morocco, then finally the adaptive trait were more mentioned as a motivation, and here the environmental and ecological value was so small in the total set of answers. So that's why we always have to see the background of the respondents. And here are some examples of the directed interviews. Well, it depends whether this is commercial or non-commercial cryoconservation, whether you make it for future business or you make it for the society, for the public good. And also, it depends on the practical possibilities of collecting. Of course, so this is the answer from the practical world of a gene bank. Sometimes you would like to collect this breed, but you can't. So that's even if you have a nice strategy. So the impact, the relationship between in situ and ex situ. So then we were quite happy because very few people considered that supporting ex situ cryopreservation would decrease in situ conservation efforts. And that finally there could be negative effects for funding because money going to ex situ, less money for in situ. This was also a very small number of answers. So the highest number of answers were that to revitalize family origins you need both, and you need conservation of within breed, decreased consanguinity. All this is both an objective for ex situ and in situ. And finally, we had a very positive outcome from these answers that there is no competition between the ex situ strategy and the in situ strategy, and this is important message because some people were asking the question that there may be competition. So, but techniques, we have techniques, practices, they are well known in the world of gene bank managers. probably here in this room, but they are not so well known by farmers. And farmers like this breeder have prior ideas depending on other opinion leaders or newspapers or what they hear in the TV. So this one's answered to my colleague. The genetic knowledge that we are trying to acquire, for me, it is to make animals ultra standardized, which will be even more favorable to an even more intensive agriculture. So this guy is not interested by XC2 or gene banks because for him, or her, I don't know, it is going together with a farming system, intensive industrial farming system that it doesn't support. And if we read what was written by this review by Lynn Grunefeld and all, then genomic acid integration could ultimately be used to introduce favorable alleles found in rare breeds into more widely used breeds. So this is a positive view of the same idea, and this is very negative. So at the farmer level, individual farmers, you have completely different understanding or opinion as compared to the scientific world on this particular issue of how we can use fancy technologies or genomics for the management of population and genetic resources. And also some farmers, finally, not only this one, said that they are not following the recommendation of the administration because they consider that these recommendations are just supporting the dominant livestock model. So maybe it's only a French business, but at least it showed that there are controversies which are hidden between this consensus. So we looked also at trade-offs regarding technologies, and this is a rather complex figures where this question. Further research on semen properties, is it supported? Yes, strongly. But here, using cloning methods to increase the number of animals of a rare breed, no, we have more no than yes. Then all the others, whether we need to kill a few females of a rare rabbit breed to obtain embryos, yes, the answer was yes. Sacrifice a few number of chicks to sample gonad for further grafting, a technology developed in IMAGE, we have more yes. And this question, which is about the mirror of this one, if this is the only option, collecting tissue samples, knowing that you will have to use cloning or reprogramming methods, then you have more yes than no, which means that if you are in an emergency situation, maybe there is a better acceptance of fancy techniques, but it should not be the initial first answer. It's more no than yes, but if they say, well, okay, I prefer to do something than nothing, so maybe I can accept this approach. And so also we collected different opinions from interviews. So a breeder saying we are being offered the opportunity to participate in semen collection to take advantage of the current technical tools, but we categorically refuse these technologies. Well, categorically. So then we have a lot of discussion to have with this breeder. And when you see a gene bank manager, it's completely the opposite. Many new developments are still to come, and I certainly believe that these innovations will trigger the use of gene bank collection in the future. So we have to deal with these very contrasted opinions because the animals we want to preserve, they are with this person. And then we want to connect with the gene banks. So we don't have an answer because that's the issue with ethics. You raise questions more often than you have answers. So the discussion that finally what we have observed comparing the interviews and the surveys, that all the challenges we are talking in the project in IMAGE and here, they are, they do not go out our world, of gene banking. In the social world of animal production, they are not perceived at all the same way as what we can discuss in our arena because we are used to these questions and we are self-convinced that it is for the sake of people, but it's not the perception maybe of these people. And our world of gene banking until now has a rather conservative view of gene banking when you do the discussion of putting in priority endangered breeds, whereas as was said just previously, thinking of preserving a breed before it is endangered is still not a common practice. And then to address these questions, Antoine proposed to identify 4 categories of stakeholders: the enabling, functional, diffused, and normative. And so that's what you find here. So we are our technical innovations, what we have been working in IMAGE. So the enabling actors, these are the legislators, the coordinators, the ERFP. the normative that the biotech service provider, the breed society, those that, well, if they don't want to do it, they will not do it, if they cannot. And then the functional, that's where we have the science, the managers, the breeders, all that really doing the work. And this is the diffused, we call that, so we may discuss what is diffused stakeholder, diffused linkage, all the NGOs or special interest groups, which are from time to time motivated, but not maybe all the time. It is more diffused concern. So the conclusion is that, of course, we have ethical implications in biobanks because they have to act and they are in relation with many different actors. And they are well known, the biobanks, only by a specialized public, and they should be popularized in order to have a better, maybe, connection with the farmers, the individual farmers. And for that, we finally considered that to get the interest of different types of stakeholders, we have to have a different speech, not to tell different things, but to find a way to attract them from their viewpoint, which means that for NGOs and farmers, we have to show it is useful for preserving their flock, not for the general world. And for breeding companies, we have to show it can be useful for their breeding program, not for the rare breed, because, okay, they say this is good, but it is not my business. And also, we have to show that these are opportunities for research, because biobanks are a driver for research in genetics and in reproductive physiology. And they are also a leverage for a global European strategy of animal genetic resources. That's what we need to communicate with decision makers. So this is an effort. We have to prepare our communication strategy tailored on the different type of stakeholders, because we have seen there are so different views from the stakeholders. So that's work is continuing, work in progress, I would say. Thank you for your attention.
Thank you very much indeed. Any questions?
I agree with you that this is what we can probably conclude from this answer, but that's the limitation of the survey. So we had a last section of the survey. Do you have any comment or so free text? And it was never used in the survey. So that's why we need to have these interviews also to get more, because I agree with you that cloning, it's reproducing identical animals, and we are speaking about diversities, so probably it is an explanation, but also it could be that cloning now you cannot have in the food chain products from cloned animals, so could be also one reason. But I agree that this was the only strong, not trade-off we could see. So, but we can continue working on it.
Good morning. What I have found the most serious concern in terms of ethical aspects of the gene are really in the use of the conserved material. How we can take the right decisions about the correct use of these resources that we have. I don't know if I'm explaining right.
Yeah.
I have found less concern when I am creating the bank than when I have the bank and I have to take a decision with respect to the use of the bank.
Yes, maybe for the question is about, I've mentioned many ethical issues at the time of making the collection, sampling, but there are ethical issues at the time of using, which were not detailed in the survey. But at least one answer that was very clear, that the governance needs to be multi-actor. So also for the decision about uses, It cannot be a single person who takes the decision. And all the skill of the gene bank is to organize the governing board, including the different profiles or the different types of stakeholders, so that it is still recognized as reliable governance. Otherwise, it could be very easily, quickly criticized to favor one, and then it takes a lot of time to go back to trust if you have lost trust. So the main recommendation from this survey, that I didn't show that question, that the multi-actor decision governance was the key to keep the trust.
Okay, thank you very much. I would like to thank all three speakers or four speakers for giving a very nice and broad intro into the topics of the day. Thank you very much.
And now we have the... Susan, we have the break.
Yes, there's a coffee break now. It's in the second floor of the main building, so you have to go out on the left and then enter the main building. In the entrance, you will find a poster, the stand, which are posters that have been produced along the project. So you can have a look. And in the second floor, you have the coffee break. We should be back at 11. Because for lunch we have a small story. We should be really starting at 12 lunchtime to be able to follow in the afternoon.